13 universal. This is A-level CIE biology with Shedro. So this is all topics theory paper 2. This is paper 2 series 2. Okay, we have done series 1. So follow us, let's dive in. Get your writing pads and pens. Let's dive in. Okay, so let's look at the first question. It says hemoglobin is a complex protein. Okay, it is a complex protein. Is a complex protein molecule made of 4 separate subunits as shown in Fig 2.1. That's it. 4 separate subunits. You can see the subunits. Okay, so these are stimulus materials. When solving biology questions, you have to pay attention to stimulus materials, the text, and then the diagrams. Okay, so these are the subunits. All right. You see, this is a subunit. This is also a subunit. All right, about 4 of them. Okay, let's go to the question. The first question says the shading represents the 2 different polypeptides that form a molecule of hemoglobin. State the names of 2 different polypeptides. Of course, you know the names. So pause the video and write down the answer. What are the 2 names of polypeptides? 2 names of polypeptides, right? 2 names of polypeptide. Don't forget, one is alpha globin. You know alpha, I can spell it A-L-P-H-A, alpha globin or polypeptide. Is that okay? Or you write the alphabet. You know the symbol? Okay? You can write the symbol like this, alpha globin. This is alpha globin, all right? It's alpha globin, all right? Or beta pleated, okay? Beta globin, not beta pletation. So this alpha globin, right? Just 2 of them, okay? Or beta globin polypeptide. All right. So globin, the same thing here, globin. All right. Okay. So very importantly, guys. All right. Let's look at what the math scheme says because we follow the math scheme. All right. So quickly, the answer: if only symbols are used, then both have to be clear. So I'm correct. So C. So you're going to use say alpha. All right. This is the letter or symbol, globin or polypeptide. And then we'll see and beta. See the symbol, globin polypeptide. All right, let's go on to the next question. So ignore alpha and beta polypeptides. No, you have to bring in globin. All right. So you say alpha globin or polypeptide. You just list them. All right. And if you're saying polypeptide, put chain there. Okay. So let's go to the next question, guys. Just 1 mark. All right. Next question. The same question there. So it says identify the structure labeled P in Fig 3.1. So see this P. What do you think is P? P is what? It should be hem group, right? Hem group. So hem group is a prosthetic group in hemoglobin and it contains iron 2 ion. What is hem group, right? So you can use this spelling is very important in biology, or HEMA or prosthetic group. Accept hem group. All right, ignore porphyrin ring, iron. This is iron 2 ion, so ignore them. All right, so that's just 1 mark. Our next question. AII Fig 3.1 shows some of the levels of protein structure. State the level of protein structure that is not shown. Okay, there are about 4 levels of protein structure. You can try this question too. It's a simple question. 4 levels of protein structure. We have the tertiary structure. Of course, that's more than 1 polypeptide chain. You can see we have more than 1 polypeptide chain here. This is a polypeptide chain. This is another polypeptide chain. This is another one. Okay, this is another one. Okay, so there are 4 polypeptide chains here. So that's quaternary. Then we have tertiary. Tertiary is a 3D structure. So this is a 3D shape. All right. And then we have the secondary. Secondary, we have this helix, alpha helix here. Okay, it's a secondary structure. All right. And then there's the last one, which is primary structure. We cannot see primary structure, which is a sequence of amino acid, and that is what is not shown. Okay. So the answer is primary structure or sequence of amino acid in a polypeptide. That's primary structure. Okay, that's not so. Just one mark. Let's go to the next question, guys. All right, it says hemoglobin is involved in the transport of carbon dioxide. Molecules of carbon dioxide enter red blood cells as the cells travel in capillaries through muscle tissue. Some of these molecules are converted to carbonic acid, right? So when carbon dioxide combines with water, catalyzed by carbonic anhydrase, forms carbonic acid, which dissociates to form hydrogen and carbonate. You know that. Explain. Explain means give us reason. Okay, how hemoglobin is involved in the transport of carbon dioxide molecules that are not converted to carbonic acid. So how is hemoglobin involved? Explain. So hemoglobin, right? Explain how. Hemoglobin binds to, of course, you can see that the carbon dioxide here, if you remember that carbon dioxide um, binds to hemoglobin, right? So it has to bind with hemoglobin. So when it binds to hemoglobin, which is Hb, um, for transport. But the question is, which part of hemoglobin does it bind to? It binds to the, of course, this is a reversible reaction. It binds to the terminal amine group or amino group. Terminal amine group, amino group. So to form what we call carbamino hemoglobin. So this is carbamino hemoglobin. So we have carbamino. That's carbamino. 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 Carbamino hemoglobin. Carbamino hemoglobin. Okay. Carbamino hemoglobin. Yes. So let me just call carbamino HB. But carbamino hemoglobin. So it binds to that. So we can have that. All right. So it binds with the terminal amine group. Okay. And you know where it binds to it because they explained how. So it binds. How does it bind to hemoglobin? It binds to the terminal amine group to form carbamino hemoglobin. Okay, and it binds at high partial pressure of CO2. That is in respiring tissue. Okay, and then at low partial pressure of CO2, it dissociates carbamino hemoglobin dissociate to give us back CO2 and hemoglobin, and that is at alveoli when there's low partial pressure of CO2. Okay, so the answer is carbamino hemoglobin. You know that. Okay, yeah. Um, so see what the math scheme says. Any three forms. See the math scheme here. Please don't forget that our materials come from past questions, CIE, all right, Cambridge. Hemoglobin combines with, you can see math scheme, the same thing, combines with or binds to or reacts with carbon dioxide. Okay, carbon dioxide reacts with terminal, so terminal amino group, that's amino group, okay, chain, okay, of each chain. So this approach, so it has 4 chains. Hemoglobin has 4 chains. Each chain of them has a reject R group. So A forms a bond. So accept A means accept forms a bond. So it forms a bond to form carbamino hemoglobin. Now you can see the rest of the, okay, carbon dioxide is bound to Hb until a region of low. Okay, that's when it releases it. So in the lungs, it unbinds or dissociates. All right. Question number 1C says, okay, when hemoglobin associates or binds with oxygen, it forms oxyhemoglobin. Yes. State the precise site in the mammalian body where hemoglobin molecules binds with oxygen. Okay, that happens in the alveolar capillary. Okay, the capillaries surrounding the alveolus. Remember, guys, okay, how this happens. Let me paint the picture for you. Okay, they say state. So this question is testing your memory. So this is the epithelial cells of alveolus. That's epithelial cells of alveolus. Okay, so these are the cells of alveolus. Okay, now this is the endothelial cell of capillary. Right, see it here. Endothelial cell capillary. Yeah. Okay. So this is endothelial cell capillary. Oh, okay, let me clean up this. All right, let's look at it. Yeah, so you can see, so remember we are solving question, but also we are learning. So you see the endothelial capillary pores and all of that. So red blood cell is somewhere here, okay, and that's where hemoglobin is. Hemoglobin is inside here. Let me call it H, okay, small h. So this is the site. So oxygen, there's high partial pressure of oxygen in the alveolus, alveolar air. This is alveolus, this is the air inside alveolus. So it's going to diffuse from high partial pressure, right, across the endothelial, epithelial, of course, cells of alveolar wall, and across the endothelial, so into the red blood cell. So the site is the alveolar capillary. That's where it diffuses into the red blood cells and combines with hemoglobin. Okay, so let's look at what the mass scheme says, but I painted the picture here. All right, so answer here says alveolar capillary around alveolar in the lungs, just 1 mark. All right, next question. Okay, so the compound 2,3-diphosphoglycerate 2,3-DPG is produced in red blood cells. Okay, it says P50 is the partial pressure of oxygen when hemoglobin is 50% saturated. The P50 is used to compare the affinity of hemoglobin for oxygen under different conditions. Okay, let's look at the graph. That's the graph. So you see at low concentration of 2,3-DPG, this is for high, right? So this is because I said that P50 is a partial pressure. You have to understand the question. Don't just jump into understand what I'm trying to say. Okay, P50 is a partial pressure of oxygen when hemoglobin is 50% saturated. So when hemoglobin is 50% saturated, the P50 is used to compare. So it is the partial pressure. This is the partial pressure, okay? So according to this graph, when is hemoglobin 50% saturated? So this is P50. If you get here, go down, that's about 3.2. okay, each box is about 0.2. so this P for this one, the P50 for the other graph is about 4.6 or thereabout. Okay, all right, so let's go on to the question. Say fig 3.2 shows oxygen saturation curves when red blood cells have high and low concentration of 2,3-DPG. Yes, we've seen that. Use the information in fig 3.2 to describe the effect of an increase in the concentration of 2,3-PG on the oxygen saturation curve. The effect. So say we should describe the effect, all right, of increase this. Can you see that? Now look at when there's low concentration, we can pause this video and try to solve it. Okay, pause the video, but you need the knowledge on this topic to solve this question. Okay, so if you check at low concentration of 2,3-PG, you can see that. Let's check the P50. The P50 here, all right, is 3.2. okay, 3.2 somewhere here. Okay, but when there's high concentration of 2,3-PG is about 4.2. Just trace it down. Okay, pause the video and trace it down. That's about 4.2, right? So they are now asking us to use the information to describe the effect. So you can see that, watch this guys. If I trace this graph, right, see that at low concentration of this, the P50, all right, is smaller. That means it combines with more oxygen. Okay, hemoglobin combines with more oxygen, all right, at low concentration, right? So the P50 is 3.2, but at high concentration, all right, the P50, that's the partial pressure at half percent saturation of hemoglobin, is about 4.2. Do you get? All right. So why is that? You see that at low concentration of 2,3-PG, what happens is that hemoglobin combines, and that is why even if you follow this, you can see that it has at low concentration of this, hemoglobin combines more with oxygen. Do you get? So you see the partial pressure, if I use 4 now, okay, the partial pressure is about 60, 66 or thereabout, okay? But at high, the same pressure is 40, okay? So you can see that hemoglobin, percentage saturation of hemoglobin to oxygen is higher, all right, at low concentration of 2.23 dpg. You can see that example here is 60 point, okay, and it is lower, right, when the 23 dpg is high in concentration. So you can see that's 40%. All right, because they were asked use the information to describe the effect, right, of increase. So if you increase the concentration, what happens is that the percentage saturation of hemoglobin with oxygen will decrease. So increase in composition. So what happens is that, how do we know that in the graph? The graph will shift to the right. Can you see it shifts to the right? The graph shifts to the right like this. All right, shift to the right. Okay. All right, and that means that it shifts to the right, but the graph is now below, right? So less oxygen combines with hemoglobin, okay, because if we check it there, at low it has higher percentage saturation, at high this it has lower. So less oxygen combines with hemoglobin. If you increase the concentration of 2,3 dpg. Okay, let's look at what the mass scheme says. All right, mass scheme says any two of these. Curve shift to the right, yes, curve is lower. Hemoglobin is less saturated at every PO, okay, partial pressure of oxygen. All right, so it's less saturated when we increase it. All right, you see what's happening. Okay, high partial pressure of oxygen requires reach the same percentage saturation as low dpg. So for us to reach it, to reach the same 50, we needed, look at 50 here was 3.2. all right, 50 for this 4.2. So we need a higher partial pressure of oxygen to reach the same saturation. All right, so as low. So can you see 50 is higher? Okay, yes, partial pressure. Okay, so use data for P. This is 3.2 here, but when we increased it, all right, from here to here, it now increased from 3.2 to 4.6. So that's the effect. Don't use Bohr effect, because Bohr effect has to do with CO2. All right, let's go to the next question. I think we're done with this question. Blood is stored in blood banks for use in hospitals during operations. The concentration of 2,3 dpg in red blood cells decreases when blood is stored in a blood bank. State and explain the effect that the use of blood taken from a blood bank has on the supply of oxygen to tissues of a person during an operation. So you see, the blood stored in a blood bank, guys, don't forget that it has not taken up oxygen. So it doesn't have a high amount of oxygen, so it's going to deliver less oxygen, all right, to the tissues. All right, it's going to deliver less oxygen to the tissues. Okay, yes, because the amount of oxygen is small, so the affinity of hemoglobin for oxygen will be low. All right. Okay, so look at it. Less oxygen is available for tissues. Hemoglobin it only has high affinity for oxygen. All right, hemoglobin has high affinity for oxygen. Now, why does it have high affinity for oxygen? All right, if the hemoglobin of this blood, remember that if the hemoglobin of this blood still attached to the oxygen at low partial pressure of oxygen, that means that hemoglobin has higher affinity for oxygen. Remember that hemoglobin always unloads oxygen at low partial pressure of oxygen, but if it still holds on to it, because this blood has been without oxygen stored in the bank, so hemoglobin is still attached to it, that means that hemoglobin has higher, okay, affinity, but less oxygen is available for tissues because it has low partial pressure of oxygen. Okay, all right, so let's go to the next question. Yeah, fig 4.1 is a diagram showing the transcription of part of col1A2 gene that codes for a collagen polypeptide. The part of the col1A2 gene shown in the section of exon structure, okay, exon structure A, represent an enzyme involved in transcription. So look at it. The enzyme involved in transcription. All right. So this is the enzyme involved in that structure A. This is DNA transcription going on. This is the enzyme involved. You can see the enzyme involved. Okay, so structure A is the enzyme that is synthesizing this primary transcript. Okay, so, and of course this is template, the transcribed strand. That's not transcribed strand. Okay, what do you think is the answer? There's a name the enzyme labeled A. What is A? Okay, which enzyme catalyzes transcription? Okay, all right, of course it's DNA polymerase, RNA polymerase, sorry, RNA polymerase 1 mark. So DNA polymerase is for DNA replication, but for transcription is RNA polymerase. Okay, so the answer is RNA polymerase. Okay, fig. Okay, that's same thing. They ask another question. Name the bond that is formed between nucleotide in the primary transcript. So which bonds are formed between nucleotides? Okay, so these are simple questions we should know. Which bonds are formed between two nucleotides? Of course, phosphodiester bond. Let's go on. All right, fig. The same question. Let's look at the next question. State the number of amino acids that are coded for by the sequence of nucleotides on the primary transcript. So this is primary transcript, right? So don't forget that 3 amino acid gives rise, sorry, 3 bases gives rise to 1 amino acid. So this is 3. This is the first 3. This gives us 1 amino acid. This is the next 3. Okay. This is the next 3. Okay. This is the next 3. Okay. This is the next 3 here. So this gives us 1 amino acid. This gives us 1 amino acid. 1, 1, 1. So let's check it. So count amino acid. So this 3 gives us 1, 2, this 3, 3. Okay, this is 4. All right, this gives us 5. 5 amino acid. All right. So that's the answer. All right. So let's look at, okay, use the information in fig 4.1 to explain why one of the strands of DNA is not transcribed. Now, why one of the strands is not transcribed? Okay, why is it not transcribed? Because it is not a template strand. This is not a template strand. Okay, so this is a template strand. So it is not transcribed because, very importantly, it carries a different genetic information, different base triplet. So it is going to code for different amino acid sequence, and it will give rise to non-functional protein. Okay, different codons, different amino acid sequence. Okay, so this is different base triplets. They'll give us different amino acid sequence that give rise to non-functional protein. Okay, and of course you can check that messenger RNA is single-stranded, so it does not need 2 strands. Okay, and to make messenger RNA, messenger RNA is coded from, is made, right, of course the primary transcript here, from 5 prime to 3 prime, because we copied the template strand from 3 prime. So template strand is copied in the direction of 3 prime to 5 prime. So this must be made from 5 prime to 3 prime. Okay. All right. So let's see what Maskim says. Any 2 from other strand is not template. Of course, we've said it. Okay. So strand is not transcribed. 1 mark. If transcribed sequence of nucleotide bases in mRNA codes for a different sequence of amino acids, right? So accept sequence of codon, yeah. So primary transcript for RNA, accept it. If transcribed result in a non-functional polypeptide, codes for a polypeptide with different function, yes. So translation will be a waste, because after it has coded for something that has no use, to be a waste, okay, translation will not occur. Any suggestion? Okay, these are the consequences for collagen there. All right, do not be, and then mRNA is single-stranded. So RNA polymerase synthesize, yes, RNA synthesizes RNA in this direction. We've said it. Okay, guys, let's go to the next question quickly. New mitochondria are formed when mitochondria divides into 2. Fig 4.1 is a transmission electron micrograph. You can see that of mitochondria that is dividing. This mitochondria is dividing, right? Is dividing. So state 2 features of mitochondria that are visible. You can see it. Look very well. You see the double membrane, the outer and inner membrane. You can see the infolding. of the inner membrane. That's crystal. Double membrane, outer and inner 1, infolding 2. Okay, you can see the matrix. See the matrix. This part here is matrix 3. So this infolding is crystal 1, double membrane, outer and inner. Okay, that's 3. Okay, that's the envelope. So quickly, not waste time. Answer: reference to mitochondrial envelope, 2 membranes, double membrane, inner membrane, outer membrane. All right. Or so reject cell surface membrane. It's not cell surface membrane. It's mitochondrial membrane. And of course, inner membrane is folded. That's crystal. We have mentioned that that's crystal. Yes, we did not mention 70S ribosomes. Yes, if you see the tiny dots, these tiny dots, they are 70S ribosomes, so they are visible also. So do not say ATS. Reject. Arrow means in mass scheme reject ATS ribosome. This ribosome is a typo. Reject ATS because ATS is not found in mitochondria. All right. So of course, mitochondrial matrix. That's matrix. Okay. So also ignore DNA. You cannot see circular DNA here. So 2 marks, any of these 2. New mitochondria are formed when a mitochondria divides into 2. You can see that the same thing we said. They said the line on Fig 4 shows the diameter of the mitochondria. Calculate the actual diameter of the mitochondria. Give you answer. So if you take your ruler, remember they have given you the magnification. You know that, and of course you know that magnification, we're looking for actual height. So if magnification is all right, image over actual. Okay, so what will be actual? Actual will be what? Actual will be of course image over magnification. So how do we get our actual? All right, okay, just take your ruler and measure from here to there. That's about 3 cm if you measure it in real paper, but here you might get, it's gonna be the same because of different font size. So 3 cm, convert 3 cm to micrometers. So 3m to micrometers. How do you work on that? Multiply by 10,000. Okay, so it will give you 30,000, right? So 30,000 micrometers over magnification. Magnification is already given to us, which is 50,000. So 50,000. All right, 50,000. 30,000 divided by 50,000. That gives you, press your calculator, 0.6. 0.6. 0.6. Okay, micrometers. Yeah, so that's the answer. All right, guys. So let's go to the next question. So 0.6 micrometers. All right. Yeah, mitochondrial DNA codes for some polypeptides or protein used within the mitochondria. Some of the proteins allow movement of ions into and out of the mitochondria. Outline the ways in which ions can move into the mitochondria, right? So how does ions move? Ions can move by active transport. Active transport is against concentration gradient from low to high, all right? So an active transport, all right, passes through carrier protein. So carrier protein has binding site, all right, for specific ions. Don't forget that it has binding site for specific ions. So carrier protein. So when the molecules bind to it, right, so what happens is that they undergo conformational change in shape. All right, you see them changing shape. So carrier proteins always undergo conformational change in shape. Okay, yeah, so you can see it has changed its shape in order to allow the ion to be transported out. All right, don't mind my diagram, okay, but you get the gist. Okay, yes, okay, it's moving out, so the ion is going out now. So you can see the ions transported, okay, out. Okay, yeah. So active transport requires ATP. These are things you're going to list. Of course, if it is channel protein involves only facilitated diffusion. Don't forget that Carrier protein is also involved in facilitated diffusion, right? But channel protein is only involved in facilitated diffusion, not active transport. Okay, so this is channel protein, so it's diffusing across. All right, it's gated hydrophilic passage. This has binding site, undergoes change of shape. All right, let's see what the masking says. All right, Na3 active transport using ATP moves ions against concentration gradients. Yeah, facilitated diffusion, passive, no ATP, no energy with, okay, of course, down concentration gradient. All right, protein carrier pump changes shape, conformation change, has binding site. If you're talking about protein carrier, channel protein for facilitated diffusion only. Okay, yeah, hydrophilic pores for ionophores. Okay, all of that. So, all right, let's go to the next question. Thank you very much, guys, for hanging out with us. So we are gonna be doing series 3. Okay, so hope you liked the class today. We want feedback from you, so share the video, send us your questions, and like the video. Thank you so much. See you next class. Bye.